At USAMRIID, the U.S. military's top infectious disease lab an hour from Washington, scientists work on some of the worst germs in the world. Anthrax, Lassa, Marburg. They're studying microbes that could be used as bioterror weapons. But the Ebola outbreak has focused their attention, says researcher Travis Warren.
I think our machines have really been running 24/7 since about June when the Ebola virus really came to prominence.
As one of the few places that can work with the Ebola safely, USAMRIID has helped develop some of the most promising drugs against it. One, called TKM Ebola, fights the disease in a novel way by targeting the virus's genes carried on a strand of RNA. The genes contain instructions that force an infected cell to make more viruses. TKM Ebola contains a small piece of the virus's RNA wrapped up in a package that alerts the cell's defenses.
There are enzymes that literally chop up the viral RNA and destroy it. And so once the viral RNA is destroyed, the virus ceases to function.
TKM Ebola is still in early testing, but USAMRIID also helped develop other, less exotic drugs that are starting or about to start human testing in West Africa.
So we have a number of products that are commonly in use for influenza virus, HIV, hepatitis C virus, and now we're starting to focus on using those types of therapies for Ebola virus.
One of these, called favipiravir, is already approved in Japan for flu. The humanitarian group Doctors Without Borders is testing it in Guinea. ZMapp, an experimental drug grown in tobacco plants, was also in part a USAMRIID project. It's based on the proteins our immune systems use to fight infections, says the Institute's viral immunology chief, John Dye.
We've taken a cocktail of 3 of these antibodies, 3 proteins that are known to fight off the virus, and put them all together. And then we inject that, and that helps control the virus.
Tests are about to begin in Liberia. But Ebola survivors already have those virus-fighting antibodies in their blood. Dye is also working on another study to try using survivors' blood to treat the sick. Meanwhile, researchers elsewhere are using machines to test the Ebola-fighting potential of drugs now used for other diseases. Mount Sinai microbiologist Adolfo Garcia Sastre spoke to VOA by Skype.
If one finds a good inhibitor in this set of drugs that are already approved for human use, then it's much easier to move it quickly into the use in humans because there is already known safety profile of the particular drug.
Sastre and his colleagues have found more than 50 that so far look promising. If any are found to slow the epidemic, advocates say the next questions will be how much is available, how much it will cost, and who will pay. Steve Baragona, VOA News, Washington.